Homeostatic sleep pressure is the physiological accumulation of the need for sleep that increases progressively during wakefulness and dissipates during sleep. This process is molecularly driven by the mitochondria of sleep control neurons, specifically those projecting to the dorsal fan-shaped body, DFBNs, where an overaccumulation of adenosine triphosphate, ADP, and subsequent electron leakage create an oxidative signal that induces neuronal hyperexcitability, structurally forcing the organism into sleep to allow for organelle repair, lipid replenishment, and mitophagy. The biological necessity of sleep has remained one of the most enduring paradoxes in evolutionary neuroscience. While sleep is universally required across animal phyla, the fundamental homeostatic mechanism that senses and records the duration of prior wakefulness, biochemically termed sleep pressure, has historically been viewed as a brain-wide network phenomenon mediated strictly by extracellular neuromodulators like adenosine. However, cutting-edge single-cell transcriptomics and molecular imaging have overturned this model. Breakthrough research demonstrates that the homeostatic sleep homeostat is not an abstract systemic calculation, but is instead a cell-autonomous, bioenergetic process localized within the cellular powerhouses of specific sleep control neurons. In complex organisms, sleep pressure is directly encoded by the metabolic and structural dynamics of mitochondria inside a specialized subset of cells, specifically the neurons projecting to the dorsal fan-shaped body, or DFBNs. These neurons operate as the brain's intrinsic metabolic switch, gating sleep-wake transitions based on the precise accumulation of mitochondrial respiratory intermediates. When an organism is awake, the brain is subjected to intense and prolonged energetic demands required to orchestrate synaptic plasticity and process sensory inputs. Paradoxically, single-cell transcriptomic profiling reveals that sleep deprivation does not cause a generalized, brain-wide upregulation of metabolic genes. Instead, this transcriptional response is highly localized. Following prolonged wakefulness, DFBNs exhibit an almost exclusive, selective upregulation of genes encoding proteins critical for mitochondrial respiration and ADP synthesis. This cell-specific genetic reprogramming points to an anti-cyclical relationship between global energy availability and localized cellular consumption. During waking hours, when caloric intake and systemic glucose levels are high, the electrical activity of sleep-inducing DFBNs is actively suppressed by arousing neuromodulatory inputs, particularly dopamine. This dopaminergic inhibition creates a profound bioenergetic mismatch inside the DFBNs. Because these neurons are electrically quiet during wakefulness, their internal consumption of energy drops, causing their intracellular reserves of adenosine triphosphate, or ADP, to become heavily saturated. Quantitative assessments utilizing genetically encoded ADP sensors confirm that prolonged wakefulness drives a significant rise in ADP concentrations specifically within DFBNs, a phenomenon not observed in surrounding projection neurons. This accumulation of ADP stalls the normal kinetics of cellular respiration. Under normal conditions, mitochondria generate ADP by passing electrons through complexes 1 through 4 of the electron transport chain, generating a proton motive force across the inner mitochondrial membrane that drives ADP synthase. However, when the intracellular ADP to ADP ratio becomes excessively high, ADP synthase is structurally inhibited by the lack of substrate. This inhibition stalls the proton pump, leading to a massive increase in the proton motive force and an overfilling of the upstream mobile electron carrier, the coenzyme Q-pool. As a consequence of this electronic logjam, electrons entering the respiratory chain from a high NADH to NAD+ ratio can no longer flow cleanly to oxygen at complex 4. Instead, they leak prematurely from the overfilled coenzyme Q-pool, engaging in non-enzymatic, single-electron reductions of oxygen. This electron surplus directly induces the intracellular overproduction of reactive oxygen species, or ROS, within the DFBNs, transforming metabolic exhaust into a precise, cumulative measure of waking time. The accumulation of reactive oxygen species inside the DFBNs serves as the primary biochemical trigger that converts a metabolic state into an electrical signal. This conversion is mediated by hyperkinetic, a specialized beta subunit of the voltage-gated potassium channel known as shaker. Hyperkinetic is an atypical aldehyde reductase that functions as an intracellular redox sensor via a stably bound nicotinamide adenine dinucleotide phosphate, or NADPH, cofactor. The oxidation state of this cofactor directly reflects the rate of electron leakage from the upstream respiratory chain. When wakefulness is prolonged, the continuous surge of mitochondrial ROS oxidizes the NADPH pool to NADP+. This redox shift alters the biophysical properties of the hyperkinetic shaker channel complex, reducing its potassium conductance. Because potassium channels are responsible for maintaining the resting membrane potential and silencing neuronal activity, the closing or inhibition of these channels causes the DFBNs to depolarize. Consequently, the sleep control neurons escape their waking inhibition and shift into a state of intense, hyperexcitable spike discharge. This electrical firing pattern actively suppresses the wake-promoting circuits of the brain, structurally forcing the organism into a state of sleep. This elegant bioenergetic feedback loop demonstrates that homeostatic sleep pressure is fundamentally driven by a deliberate mismatch between electron supply and ADP demand. To confirm this causal link, researchers successfully manipulated sleep architecture by uncoupling electron flux from ADP synthesis. By introducing mitochondrial uncoupling agents, which dissipate the inner membrane proton gradient without generating ADP, they effectively cleared the electronic logjam in the coenzyme Q pool, suppressing ROS generation and completely relieving the behavioral pressure to sleep even in sleep-deprived states. Conversely, exacerbating the bioenergetic mismatch by using a light-driven proton pump to artificially power ADP synthesis in the absence of ADP immediately precipitated an intense, unavoidable sleep state. Crucially, this metabolic stress inflicts severe structural consequences on the mitochondrial network itself. Under the weight of prolonged wakefulness and unmitigated oxidative stress, the mitochondria within DFBNs undergo profound morphological remodeling, shifting away from an elongated network towards severe mitochondrial fragmentation. This structural transition is driven by the activation of mitochondrial fission proteins, such as DRP1, which physically constrict and divide the organelles. This fragmentation is accompanied by a dramatic upregulation of mitophagy pathways to segregate and destroy the most severely uncoupled components. Furthermore, the fragmented mitochondria demonstrate a massive increase in physical contact sites with the endoplasmic reticulum, forming specialized membrane junctions known as mitochondria-associated membranes, or MEMs. These structural conduits are rapidly assembled to facilitate the non-vesicular transfer of lipids from the endoplasmic reticulum to the outer mitochondrial membrane, a vital physiological process required to replenish and repair membrane lipids that have undergone extensive lipid peroxidation during the waking oxidative surge. The dynamic structural rearrangement of the mitochondrial architecture is not merely a passive byproduct of metabolic fatigue. It actively encodes and modulates the sleep state. Genetic manipulations that artificially force either mitochondrial fission or fusion within DFBNs alter sleep duration and neuronal excitability in opposite directions. Forcing hyperfusion by suppressing fission machinery or overexpressing fusion proteins like mitofusin or OPA1 significantly amplifies the electrical excitability of these sleep control neurons, causing a profound extension of total sleep time. Conversely, forcing mitochondrial fragmentation through the genetic activation of fission factors completely suppresses DFBN excitability, rendering the organism highly resistant to sleep. These findings confirm that the physical structure of the mitochondrial network operates as a direct rheostat for neuronal firing and behavioral homeostasis. Once the organism enters the sleep state, the bioenergetic dynamics of the DFBNs undergo a complete reversal. As the rest of the brain quietens, the dopaminergic inhibition on DFBNs is lifted, allowing them to fire continuously. This prolonged spike discharge rapidly consumes the accumulated intracellular ADP stores, reducing the high ADP to ADP ratio. As ADP reserves are depleted to fuel the energetic costs of sustained neuronal firing, ADP synthase resumes high-capacity operation. The proton motive force collapses back to physiological levels, and the electronic logjam within the coenzyme Q pool is relieved. Electrons once again flow cleanly and enzymatically to oxygen at complex 4, instantly halting the premature electron leak and bringing ROS production to a stop. With the oxidative stress terminated, the cellular environment shifts toward a reductive state, converting NADP plus back to NADPH. This metabolic clearance restores the full conductance of the hyperkinetic Shaker potassium channels, gradually hyperpolarizing the DFBNs and easing the organism back toward wakefulness. Concurrently, the structural damage inflicted during wakefulness is repaired. Mitochondria dissociate from the endoplasmic reticulum, mitophagy pathways subside, and individual mitochondrial fragments undergo fusion to reconstruct elongated, highly efficient networks capable of supporting the next cycle of wakefulness. This paradigm-shifting research establishes a direct molecular link between circadian biology, metabolic flux, and the evolutionary origins of sleep. It demonstrates that sleep did not evolve merely as a high-level cognitive behavior for memory consolidation, but rather emerged as a fundamental, cell-autonomous metabolic requirement to resolve the electron imbalances inherent to aerobic respiration in highly active cells. Subtle chronic impairments in mitochondrial quality control or disruptions in the structural dynamics of fission and fusion can directly distort the homeostatic sensing of sleep pressure. This insight explains the pervasive, bidirectional link between chronic sleep architecture fragmentation and the accelerated development of neurodegenerative and systemic aging pathologies. By identifying the mitochondrial respiratory chain as the ultimate origin of the sleep switch, modern longevity medicine is handed a precise set of molecular targets. Interventions designed to optimize mitochondrial membrane potential, preserve the fidelity of electron transport chain complexes, and protect against lipid peroxidation offer a revolutionary approach to stabilizing sleep homeostasis, dampening systemic inflammaging, and extending human health span at its deepest bioenergetic source.